I'm Sarah Fitzsimmons. I'm the Director of Restoration with the American Chestnut Foundation. Welcome to Chestnut Chat 4. We've had 3 excellent sessions. I see a couple people chat about how much they enjoyed last week's session on transgenics, and we invited our partnering scientists from SUNY ESF to give a great talk. We had one quick question about whether or not that chat recording has been posted. I think that was done just last night, and Jules will be sharing that link shortly. A couple of housekeeping things. We've got 130 attendees so far. You guys are all muted. You won't be able to speak or share your screens. So if you have questions, if you need something, please use the chat box. If you have a question, please use the Q&A. We will either answer questions by typing it in and you'll be able to see it in the Q&A, or we'll choose to answer it live, and we'll ask our Meadowview research farms to take those questions. We're going to hold all of those questions until the end of the session. So the formal part of our presentation will last about half an hour, and then we should have about a half an hour to answer questions. So with that, I'm going to turn the reins over to Lisa Thompson to give an introduction to our Meadowview research farm field team.
Good morning, everybody. I'm Lisa Thompson, President and CEO of the American Chestnut Foundation. I'm really delighted to have our team members from Meadowview Research Farms today. We tried to figure out how to get you there virtually via GoPro and all those kinds of things, but they were, they're very busy right now with a, kind of scaled down crew without volunteers. So we're just grateful they took the time, and they're going to be sharing some photos via the share screen process. So I won't take much time except to just thank you all for joining us. We've really been pleased at the turnout of the Chestnut Chats. We have 143 participants already today. That's fabulous. And with that, I'm actually going to turn it over to Dan McKinnon. He is our Meadowview Research Farm Operations Director, and he really manages all the activities on the farm. And he will introduce his team as he pulls it up on the screen right now. So good timing. Thank you, Dan, and take it away.
Eric's running that. Thank you very much, Lisa, and thanks everybody for joining us. We greatly appreciate your interest in our program and the interest in Meadowview Research Farms. It's been my pleasure and honor to work here for the last 6 years, as is Eric. We've been a pretty good team there. Getting a lot done, even in the wake of this global pandemic. We've just made some very minor changes, I guess you would say, but things are going on generally as normal as they would be on the farms, other than we just have a little bit different scheduling. We don't get to see each other as often, of course, where everybody's very experienced in what they're doing, so it doesn't take a whole lot of direction to keep the thing keep the operation rolling. So I greatly appreciate my very professional and outstanding team there at the farm, as well as all the support that we get from the national office, our RSCs, Sarah and Kendra. It's just been a tremendous experience working in Meadowview. I almost said here in Meadowview, but of course I'm at my house, which is one of the things that have really changed on the farm recently, is that we're doing a lot of administrative work from home, just following CDC guidelines. If you don't have to be a physical presence on the farm, we're just not showing up to the farm for emails and stuff like that. So, of course, just going over a couple of the things that have changed, we're reducing our backcross breeding season. We're going to be doing a minimal amount of that this year. Of course, as Lisa mentioned, our volunteer activities have been canceled. We very much appreciate the tremendous amount of support that we get from our volunteers, but in the light of safety in this changing environment, we've just decided to not have groups or visitors onto the farm for everyone's safety, of course. Just a quick introduction of myself, I guess. Of course, I'm Dan McKinnon, the Director of Farm Operations, but I have a very supportive team. My job there is more or less budgetary management, just kind of supervising the flow of activities on the farm. I do a lot of training and safety management and that sort of thing. Work closely with all of our collaborators. and the national office, of course, to enforce company policies and help change. We've changed a lot of company policies over the last few years, helping manage logistics on the farm, which of course is a pretty big job. As Eric's going to show in his presentation, we have a fairly large operation that is spread out throughout the community of Meadowview. We have 4 farms there in Meadowview, which is in Southwest Virginia. If some folks that are attending today have not been to the farms, once travel restrictions are lifted, we look forward to seeing you. We love giving tours of the operation. Eric handles most of those, but I think all of us have given a tour at one time or another, and we enjoy every one. So as I said, once travel restrictions are lifted, please reach out to us and come by and see us, whether it's to help out with the work there or just to become more familiar with what we're doing in the operation. We look forward to seeing you. A little bit of backstory, real quick, just to introduce myself. I got interested in the American chestnut by pretty much working for my grandfather. He started me working at a very young age, and we love to go out and just drive through the country and find American chestnut trees. And pretty much on a weekly basis, he told me the story of the American chestnut. And we would travel around, we would reclimate lumber. A lot of people would call him up and say, hey, I've got a big pile of American chestnut in my barn. I'm sure a lot of you have similar stories to that. And we would reclimate that wood into furniture and mantelpieces and that sort of thing. And I really didn't understand that there was a restoration effort as a kid. And once I got out of the Marine Corps, I came back to Southwest Virginia. I started working with Department of Conservation and Recreation in Virginia, Hunger Mother State Park, the United States Forest Service here at Mount Rogers. And eventually somebody mentioned the American Chestnut Foundation, and it was right about the time a job opening in Meadowview popped up. And I jumped on that as quick as I possibly could. I recognized this as a very unique opportunity, especially in this area. We're kind of right in the center of the native range. And I was shocked to find out that there was a restoration effort for, I guess, what you would consider my favorite tree. So I'm very honored to be working in this organization and with all of our collaborators, and especially with my team there in Meadowview. And with that, I'm going to turn it over to Eric Jenkins, who has been our tree breeding coordinator. It's been my pleasure to work with him for the last 6 years, as I've said, and he's put together a wonderful presentation for us today. So take it away, Eric.
Thanks, Dan. I appreciate all your kind words about myself and the rest of the team. As Dan said, I'm a tree breeding coordinator, and I work a lot, obviously, on the tree breeding, but also harvesting and managing like plantings of progeny tests and things like that. I got interested in chestnut. It's probably a similar story to most of yours. My dad, when his family and he's this little kid, moved into a very rural area, and before they could build the house that my aunt still lives in, there was like a shack that they stayed in that was made out of American chestnut, and they had to stay in that for a while until they could get their home built. And of course they had some Chinese chestnut in their yard like most of us do. And so when, actually it was in a pasture, so if you wanted chestnuts, you had to run out there and get them before the cows did. And so then from that, they started talking about American chestnut and telling me how, they used to be very large, and my grandfather would tell me about that. And then I think what really stuck with me was once me and my dad were out hunting, and we hunted together when I was in my teens, and he found a chestnut that had some burrs on it. And most, it's pretty rare to find one. He was really excited. And if you knew my dad, he doesn't get excited easily, or at least he doesn't show it. But he was like, hey, look at this, So that's something I really remember from when I was a kid. That's how excited he was to find one. And it wasn't very big, but it was really fascinating to him. And so I started working out here just a few months before Dan did, and I really enjoyed it, and the people I work with. And I guess that's it for me. I'd like to let Lily introduce herself now.
Thanks, Eric. Hi, my name is Lily Kingsolver. I'm the newest member of the farm team. I just started full-time in January. I've been interested in chestnuts and kind of aware of chestnuts since I was really small. I grew up in Meadowview, so I always knew that the Meadowview research farms were there. And then in college, for a couple summers, I interned at the research farms. After I graduated from the University of Virginia, I worked at Hungry Mother State Park as a naturalist interpreter for a couple of years, but I just couldn't stay away, so I started full-time at the research farms in January, as I said. And so I've been working as the nursery manager. I've been working a lot with the young trees, kind of guiding them from seed to whatever they're destined for, which we have lots and lots of baby trees right now that are destined for different things that I think Eric's going to talk about in his presentation. I am really thankful to be at the research farms. It's a really great place with a really supportive team. They've been teaching me a lot. I've been learning a ton, and I've really enjoyed it so far, and I'm really excited for the rest of my first full breeding season. With that, I will pass it on to Brandon.
Thanks, Lily. Hi, everybody. My name is Brandon Yanez Breeding. I've worked for TACF at the Meadowview office now for about 5 years. I graduated from Virginia Tech with a degree in landscape architecture. Shortly thereafter, I stumbled into TACF, really kind of accidentally. I just saw a public presentation one day, and next thing I know, 5 years have passed. So my introduction to chestnuts is essentially 3 decades in the making. I was more or less raised at my grandparents' house where they had several large Chinese chestnuts. So I have memories from being in diapers, sitting on the rooftop with my grandparents eating chestnuts that had fallen onto the roofs. And so I'm actually fortunate enough to have a section of that stump in my office at Meadowview. And so every day I go to work and I kind of get to look at that artifact, and that for me is really the main reason why I work for and with TACF. Is it's a nostalgic aspect for me. It's a way to kind of reconnect to my roots and my heritage, and to do some really kind of meaningful and hopefully very productive work in the process. So it's just really been an interesting and unique experience working for TACF. I work in the Meadowview office on the ground level, so I'm the kind of boots and gloves, a part of that team. So if you get chestnuts or if you send seedlings or anything to us, it's quite likely I've handled them at some point. So I'm very kind of fortunate to be involved directly on the ground level with a lot of the work that's done for TACF, from mowing and maintaining fields to seed distribution to sowing seeds and harvesting. I really appreciate the kind of well-roundedness to my involvement with the operations going on in Meadowview. That's kind of my story. I guess I'll pass it on to Jim Tolton now. He's another one of our technicians here at Meadowview.
Hey, Jim, you're muted. Jim, you need to unmute yourself and lower left-hand part of the screen.
Okay.
There you go.
Can you hear me?
There he is.
Okay. I'm Jimbo Tolton, maintenance technician. I've been at the research farm for 3 years now. But prior to that, I went to Radford College and majored in park and recreation, and always wanted to work outside. I worked for the Park Service down at Cape Hatteras for 5 years. I also worked for the Forest Service out in Granby, Colorado for a couple years. I never got on full time, so I ended up going into the manufacturing business and worked for Reynolds Metal / Ball Corporation for over 33 years. But Dan gave me an opportunity after I retired from Reynolds / ball to come to the farm and be able to work outside, which I love to do. Also, one time I planted white pines, got a contract with the Forest Service and planted over 14,000 trees. It turned out to be quite an episode. But I mean, I've loved working at the research farm. I've got to do a little bit of everything from cutting down a lot of trees that were no longer, we didn't need any longer, from bush hogging to building a pollination room in the back of the office. I've got to pretty much do a lot of different things, which makes it a lot interesting work. Plus, also just to be able to work outside. I mean, after working in a plant for, like I said, 33 and a half years, it's great to work outside. And it's also nice to know that we're on a mission to try to restore chestnut trees, because I love to hunt and fish, and I love being outside. So I appreciate the opportunity to work there, and hopefully I'll make a difference.
Thanks,
Dan?
Jim. Oh, yep, and thanks everybody. So I think what we'll do then is turn it over to Eric. Are you ready to do the formal presentation now?
Yes, I hope so.
All right.
So I guess everybody can see that we have a very fine team. I've really enjoyed working with all of them. And now I'm going to give hopefully a short presentation on some of the things that we do here at the farm. If I can get this presentation to advance. This is a map, overhead map of where we are, and the Price Farm is where I'm at now in my office. That's where our main offices and greenhouse and barns are. Just across the road is the Bryan Farm, and if you can see at the upper part of the map is the Wagner Farm, and then there down towards the middle right is Duncan Farm. Wagner and Duncan Farm are our seed orchards. This is a view of, you can see in the distance, our greenhouse, office, barns, operations building, and most of the landscape here is land part of the Price Farm. But what I want to talk about today is one of our seed orchards, which is the Duncan Farm, just about a mile or so as the crow flies. And I want to discuss, if any of you know, we planted these seed orchards over the course of probably a decade or more. You can see this is a very recent picture of the seed orchards, or of this seed orchard. But you know, originally all these trees were planted on a 1 foot spacing. There was somewhere between 20 and 30,000 trees planted and tested. And we have these remaining, just a few more to go before we've removed all the ones with the lowest levels of blight resistance and retained the ones that have better resistance to the blight. This is, these two are actually a couple of nice trees on the farm. You can see they still has a canker there at the base, but that's one of the better trees that we have. We know this tree has some significant level of blight resistance. But as time goes on, the trees tend to look like this. And if you've been keeping up with the journal, read the recent issue of the journal, or seen how we were doing with the breeding, we're getting like an intermediate level of blight resistance in these trees in a seed orchard. And usually they stay fairly healthy, produce seed, but often the bark becomes very crinkly and, as they call it, cruddy bark. As you can see here, as the canker continues to grow on the tree. So they have a better than American chestnut, obviously, but don't seem to have quite the resistance of Chinese chestnut at this time. And the reason for that is, you know, as you may know, we found that there are a lot more genes involved in the blight resistance response than we originally thought. So now I'm going to speak a little bit about the relatively new greenhouse we have on the farm. This is a view of, we have a greenhouse is the farthest one, and then a couple of shade houses there. Excuse me, I was checking the chat. This is a recent picture just after we sown several seeds in the greenhouse. Most of these seedlings will be used for the small stem assay, which I'll speak about in a minute, and most of the remainder will be planted in the field. As a little bit more recent, you can see there that the seedlings have a little tag on them, and that's a little bit difficult to see, but there's a barcode on there. So each seedling, you can just scan the barcode and then record your data on how that seedling is doing after it's inoculated. We had some plants in the greenhouse last year that were nasturtium, like attractive flower. They were to attract like aphids and insects away from chestnuts, and just hopefully do some, I guess you call it pest management that didn't involve the use of chemicals. Lily found recently this bird's nest. I don't know what species this nest is. I can sometimes tell birds from their calls, but I can't tell them from the color of their eggs. So this is the small stem assay, and you can kind of see from the size of the stem why we call it that. It's much different than a field trial where you grow the trees up for several years before you inoculate them. These is just a few months, and you make a small wound in the stem as shown here, and then you place a small bit of inoculum containing chestnut Cryphonectria parasitica onto the wound, wrap it with the parafilm, and then wait and see how the seedling responds to blight. And after a few weeks or months, you start to get something like this. You can see the distinctive orange color of Cryphonectria parasitica. And so most of the seedlings will look similar to this as the fungus starts to develop in the bark. Here we have some a recent purchase. People want to know where some of the funding goes for the foundation. Recently purchased this lift. We can tow these lifts behind the tractor. I believe that's Jim up there in that lift. He's harvesting birds. We use this during the breeding season to pollinate trees, and also during September, October harvest season. No one appreciates these lifts more than myself, because a few years ago I fell off a ladder and injured myself on the farm. I'm recovered fully now, but only lingering side effect is that I'm about a 1/4 inch shorter than I was because I compressed a vertebra in my spine. So I really appreciate having these lifts. I feel much safer. This is a new tractor and a new dump trailer. The dump trailers, we remove a lot of trees, as Jim mentions. Most of the trees that we plant end up being removed, usually only retaining about 1%. So this makes it a lot easier. We can use this trailer to haul this into a pile. We have to burn all of the trees that we remove. So using this to haul them and then dumping them out is a lot easier than transporting everything and laboriously handling it twice as you move it from seed orchards or somewhere on the farm to the burn pile. Same tractor, but this has got a attachment on it, ripping a line there for planting seedlings. I believe this is in another seed orchard. And finally, I think either Brandon or Dan took this picture, and I just really liked it. It's a chestnut seedling there. You can see the buds there, but I thought that was a really fantastic photograph, and kind of a dramatic scene in the background is the Price farm. We don't get as much snow as we used to, but we did during this year. So I appreciate your time, and look forward to questions. You can ask questions about anything, not just the things that were in this presentation. Thank you.
Thanks, Eric. That was great. And also Dan, Lily, Brandon, and Jimbo. Really appreciate you guys making an introduction of yourselves and what you all do at the farm. So yeah, I've got a couple questions from the chat, a couple questions in the Q&A. As a reminder for all of our attendees, if you have a question, we prefer it in the Q&A because then we can keep the questions and answers together. So I'm going to go to the one in the chat that was first so that I don't lose sight of it. Scott asks, can you explain any pruning regime that is used for retained stems?
We don't really do a whole lot of pruning in Meadowview. There's some Chinese trees that kind of reach out over the road. Those are about the only ones I prune. But anytime we open up a wound, of course, we expose it to a blight infection. So we just try to mitigate any pruning whatsoever. I'm not sure if you're talking about smaller stems or something like that, but we generally don't prune chestnut trees in Meadowview.
Well, a question kind of along those lines. I concur. I mean, on the orchards that I work with and maintain, we generally don't prune. We generally don't recommend it because, to your point, it does open up for a place for blight infection. But let's say you have a sprout. You're growing wild Americans, which several people on this call grow wild type Americans, which are going to get the blight regardless. It's going to die back. It's going to throw up a sprout clump. What do you guys do to help maintain those clumps, if anything?
Well, I think they used to do some of the mud packing on some of the American chestnuts when they had a blight canker. We haven't done that in a while, but if you pack mud around a canker, the organisms in the soil will definitely slow down the spread of the canker and give it a chance to recover. So if there was a specific tree that, say, you might want to use in the future breeding program, that would help it have a better chance of being used. But we haven't really done any systematic control of infection on American chestnut trees. We do have a lot of pure American chestnut trees on the farm that we maintain. We don't have a specific regime that we use for each of them.
I know some people ask me a lot, do you cut out the dead stems? Do you?
No.
That's usually what I say. The less is more. Just kind of leave them be, and sometimes that's the best thing for the tree.
Unless they're, a hazard.
Yeah, right.
Yeah, unless it's absolutely necessary, we just don't cut.
Yeah, good. Let's see. Steve Johnson Ball asks, have you investigated lac balsam as a pruning sealer?
I don't know what that is.
I don't either. And since we don't prune, probably not, but might be something interesting to look for. Let's see. Let me go over here. David Hunter asks, are all the trees that Eric showed in the orchard, are they Restoration 1.0?
Well, I think that term, we don't use that very much, but I think at one time, the seeds that came out of that orchard would have been called Restoration 1.0 seeds. If you are a member of the foundation and receive seeds, they might have come from that orchard.
Yeah.
All right. I'm just trying to organize some of these in some semblance. Let's see. All right. Eric, you mentioned that there are lots of genes for resistance, but last week during the GMO talk, it was mentioned that there was only one gene being inserted. Could you clarify?
Oh, I guess maybe I wasn't as clear. In a Chinese chestnut, there are many genes that are involved in the blight tolerance response. We don't know exactly how many, but it's much more than the 2 or 3 that we originally suspected. Now, in the transgenic tree, they insert one gene into American chestnut, and that single gene seems to produce a extremely good response to a blight infection and create blight tolerance. But in our hybrid trees, there are multiple genes involved, and specifically in a Chinese chestnut, there seems to be maybe 10 or more is what the suspicion is. And I think in that recent New York Times article, Dr. Powell had identified at least 6 that seem to affect the Chinese tree's response to blight.
I think one thing important that's noted is, though we don't know how many genes control it, it's a lot. And we've seen evidence that there may be some control on every chromosome as well. So, it's really hard to say there are X number of genes that control resistance. I think we can pretty safely assume it's a lot. What's that?
Yeah, I was just spitballing. We know it's many more than just 2 or 3, multiple times more.
Yep. All right, so I've got a couple questions about the interplay between Transgenics and the backcross program. So I want to lead this in by pointing folks, there's a question in the Q&A under the answered. Someone asked for links. There's a link to the 3-burr document. And if you guys have been following our Chestnut chats, we've talked about 3-burr a couple of times. That's the breeding, biotech, and biocontrol united for resistance. That's the document and the method that we're following to blend these 3 projects together for restoration. So we're definitely working with both traditional breeding techniques and the transgenic techniques that you learned about last week. So I don't know, Eric, Dan, Lily, Brandon, let's see, assuming the government has approved transgenics, you have transgenic pollen, you have flowers that are ready for pollination, what do you do next?
Here on farm, we intend to cross some of our better hybrid trees that are mostly American chestnut genetically with the transgenic tree. And we have done that, under very controlled conditions. We have an extensive permit process to use that pollen. And if you were participating in that, about half the trees end up inheriting the OXO gene, the transgenic gene. Looks like we have another visitor there, Sarah.
We need to put her on the payroll. She attends all these meetings. So.
So along those same lines, can the blight resistance from the backcross generations at Meadowview be combined with the blight tolerance of the wheat gene in Darling? Evan goes on to say, I'm wondering if the blight resistance can be somehow pyramided to make a stronger resistance complex.
And that's what we're looking into now, to see if comparatively, it's like, say you have a tree that has the OXO gene that's pure American. We want to compare that to see if there is a stacking effect by crossing that using that transgenic pollen on our hybrid trees. But we haven't got to the point where we can test that yet. We just now recently used the transgenic pollen, so it'll be a while before we can tell if there is an improvement. We hope there is.
Thanks, Eric. I see someone else says hi to Parker from another 5-year-old. Let's see. Is the age of the sprout or height of the sprout best used for ideal transplant time from pot to ground? So I've got a couple questions of this. So a lot of people looked at those great pictures you had of the greenhouse. When do you make that move from pot in the greenhouse to in the ground, and how do you make that happen and with the best success?
Well, we have been using the pots in the video they're about roughly a quarter of a gallon, and kind of part of the reason we use those smaller pots is because we have to plant thousands of trees, and so if you use a much larger pot, that means a lot more work to get it in the ground. So we usually, I couldn't say exactly if that's better or worse. My feeling is if you had a slightly larger pot, you end up having a larger seedling to plant, but it would be a lot more work to get it in the ground, but it may do better. It's got a more extensive root system. We have been trying to plant in the spring and trying to start our seedlings, March, in the greenhouse, and like overwintering them in another building and then planting them in the spring. So I don't know if that answers the question. We may end up planting some in the fall, but I think it's better to plant in the spring. But if somebody just had a few trees to plant, I would kind of recommend growing them in a little bit larger pot than we have, maybe a gallon, something like that, and then just planting from there. I think you'd have a little bit better success. Although we do have very good with these smaller seedlings.
Yeah, and just to kind of piggyback, Eric, so here in Pennsylvania, we grow our seedlings in pots. We usually start the seeds in January because we have greenhouse facilities to do that. That gets us a pretty nice looking seedling by late May, early June. We've got a frost coming in tonight. It's going to be 27 tonight. So we try to keep our seedlings in pots until late May. May 27 is the official frost-free date here. So that's usually when we put it in the ground is May, depending, we watch the weather real close, but yeah, we can get frost pretty late into May.
And to add to that, kind of an anecdotal observation, I just got a bunch of trees out that we grew in our nursery facility last year throughout the growing season, and then we put them into our ops building, which is a climate-controlled environment. We kept it about 40 degrees in there, and when we got the trees out, I believe it was last week, maybe the week before, they had just done spectacularly. I mean, it's kind of an anecdotal observation, of course, but I look forward to planting those out because they're extremely tall compared to the ones that were kept in the greenhouse, or we used to box them up in wax lined boxes and that sort of thing, which really stunted the growth. So storing them in our ops building this year really yielded some very positive looking results anyway.
And a lot less mold than when we stored them in.
So much less. They're so much healthier looking.
Yeah, really nice.
I got a couple of questions about Transgenic. So if you guys didn't catch last week's Chestnut Chat, there's a ton more information that's just strictly on transgenic trees. And some of you may be more familiar with the term genetic modification. So they can typically be used interchangeably. But so Tom asks, what is a transgenic tree? And Charles asks, it's my understanding that the gene conferring resistance for the transgenic does not come from Chinese chestnuts. Is that true? Would you guys feel that?
Yeah, no, it does not. The gene comes from a wheat, but nearly identical gene is found in several other plants. I think the bananas, strawberries, other fruits, other plants, probably many more than we even know about. But the gene that they use does not come from Chinese chestnut. I know that Dr. Powell has tested some genes from Chinese chestnut, individual genes from Chinese chestnut inserted into American chestnut, but they don't seem to work quite as well as this gene that turns out comes from wheat. I always found it interesting that it originally tried to use a gene from frogs. So I'm kind of pleased that he ended up using a plant gene. I think people would get really excited if he found out it was a chestnut cross with a frog. Some people are less than enthusiastic about genetic modification. I guess know as much as anybody here, and I feel good about their work, and I look forward to working with them with this and as they progress using the transgenic seeds and pollen.
All right. So back to culling. Is roguing or culling based solely on blight resistance, or do you also cull trees based on phenotype, for example, branchy growth habit versus timber type form?
We would, a lot of times in those seed orchards, and I didn't mention this before, and I meant to, there's a few trees we found in the seed orchards that are like accidental outcrosses with Chinese chestnut. So they're like, rather than being 90% American, they're closer to 50%. So they have more of a Chinese look to them, but we do want to keep these. I found that a lot of them are sterile. They don't produce pollen. So they're not contaminating the seed orchard like that. But I think if a tree did exhibit just a moderate level of blight resistance and had a really Chinese have it, we might end up culling it based on that, but it's mostly based on their level of blight resistance, the phenotype, and also the model that our geneticist has created. Model.
Yeah. So what is the typical timeline from inoculation or infection of a tree susceptible to the blight to the point? So I guess if you inoculate a tree, how long does it take to die if it's fully susceptible?
If it's fully susceptible, it's probably dead by the end of the summer. If it's a seedling planted in our progeny test, a control tree, American control, most of those don't. We inoculate like roughly in June. I'm talking about in field trials, and we inoculate them, and many of the Americans, and even a few of the hybrids, if we inoculate them with a very virulent strain, they won't even make it through the summer. By fall, they're dead. Not all of them, but many of them.
Yeah, that's my experience as well. So I'm getting a lot of questions about the material that comes out of TACF to sponsor members and long-term members. So I'm going to sort of ask this a couple different ways, and then you guys can feel this how you feel best. So let me go through a couple of these just to make sure we kind of get what these are together. So do you have zero, few, or lots of 100% resistant American chestnuts? Do you want to take that one first?
Well, no tree is 100% resistant. Even the greatest Chinese tree on earth or a transgenic tree is not 100% immune or resistant. We don't have any trees in our seed orchards that are as resistant as a typical Chinese. They tend to be about as resistant or maybe less than an F1, which is a Chinese crossed with an American, which has a moderate level of blight resistance. As for the seeds that we distribute, I feel like most of those seedlings, if they're in good conditions, you take care of them, they will probably grow large enough and long enough to produce some seeds. They're not going to attain this 6-foot diameter size, and they're going to show, at the very least, they're going to show that cruddy looking bark. But as long as the cambium layer is intact, they'll still survive, but they may not look very pretty. But again, the seeds you get from us are just going to have a modest level of blight resistance, intermediate between a Chinese and American. So it's, as Sarah's mentioned, there's so many genes involved. We don't know what many of them even do. So that's why you get a continuum from very little up to closer to Chinese chestnut.
So this gets to Alex's question, what level of disease resistance are the seedlings that I receive each spring? And then what is the average lifespan of a tree after reaching maturity?
I couldn't say the lifespan. I mean, we have some trees in our seed orchards that are, I think we started planting in 2002. So they're at least 18 years old and still relatively healthy. We have trees, at least one tree on the Wagner farm that produce 3,000 or 4,000 nuts, but it's about 80% American according to the genomic data. We found that, and it was mentioned in Sarah's article in the journal, that there's a great variation in the seed orchards as to the percentage American, percentage Chinese. The ratio is somewhat broader than we expected. When we can actually, have the technology to look at it and see how much of the genome is inherited from Chinese or American, it varies from 60 up until 90, and even some, more than 90%. But again, there's a trend, more Chinese, more resistant. And that's all due to the number of genes involved, and they're being on multiple chromosomes scattered throughout the genome.
And so kind of, again, along the same lines, how long does it take you to determine if a tree has improved resistance, and how do you measure that?
Well, we measure it. Again, I'll show you the small stem assay. We've done a lot of field trials, in the field, and we've got data from that. And so now we're using the small stem assay, which you grow the trees for 3 or 4 months and then test them. And so far, when you're taking seeds and seedlings from the same mother tree seems to correlate pretty well. So if we do a progeny test, a small stem assay in the greenhouse, by the time we get the results of that, we can know that we can know something about the parent tree and how it's how much blight resistance it's conferring onto its progeny. And we know it works pretty well because it's correlated with previous data pretty well. But again, it's a controlled environment. Field trials are not as controlled, obviously. You can't control the soil type, you can't control the moisture, you can't control anything. I've seen a lot of trees, especially in our seed orchards, which is kind of a part of the problem is many trees go for like 10 years and they look great. And then they get a blight infection, and the next year, it has a canker 2 feet long on it that looks terrible. And I've had one tree on the Duncan farm. I should have showed a picture of it. For the first couple of years I was here, it was perfect. It had no cankers at all. And I thought, this is the best tree on the farm. One year I noticed, a small canker on it, and then by the next summer, it was huge. So there's so many factors involved. It's really hard to give like a definite number on how blight resistant a tree is, especially when you have things like that when they seem fine for 8 or 10 years. Trees about this size, and then suddenly it just breaks down, or you have a bad drought. Things happen. That's why it's so hard. If it was a simple 1-to-1 ratio, either blight resistant or not, or it was just controlled by 2 genes, and it would be a lot simpler. But when there's so many genes involved and so many factors involved, it's really hard to tell. I couldn't tell anybody this seed has 20% blight resistance or 30% or something like that. It's just impossible.
So let's go a little bit of a different direction. Do you spray for insects, or are there any special fertilization procedures?
In the progeny test, we usually put Osmocote on them, but we don't do much spraying for bugs. I'll let Dan take that question because I'm not involved in that much.
Well, in Meadowview, we've actually gotten lucky. Mother Nature has cut us a break the last few growing seasons, and we just haven't had any need. We monitor, using our bucket lift, we can get right up into the canopy of our breeding trees, and we closely inspect them and everything. So we haven't really had anything that really warranted investing in more bug spray or anything like that. In most orchards, especially if you're a production orchard, of course you would be spraying for Japanese beetles is probably the primary pest here in Southwest Virginia. But we just haven't had any sort of extensive damage done, especially the last few years. It's just, we've really been lucky to not get any kind of outbreak. We had 2 years of really bad drought there. I think it was in 2015, 2016, that didn't really show any extensive damage to our trees. Didn't stress them out or anything like that. We have had a couple of wet years that we've noticed a fungal rust on our leaves, but it usually comes and goes within a month. So it's just, looking at the cost benefit analysis, do you spray something like that? Well, it's not damaging the tree necessarily. It hasn't affected seed set or pollen production or anything like that. So it's just not something that we've pursued. We do spray herbicides in our progeny test and, the smaller tree plantings, simply because traditionally our properties have been like cattle grazing land. So there's a lot of Johnson grass and stuff like that that grows up in the rows. So it's just a constant battle against the competing vegetation against those progeny test trees. And of course, when the science team comes in to inoculate, you need to be able to at least get to the stem of the tree. So we try to keep everything closely mowed and sprayed and all that, so that the tree has the best chance of survival that it can get, at least until we come along and inoculate them.
So we're almost at 12:30. We're going to go for another 8 to 10 minutes. Maybe we'll go a little over. Thank you everybody for asking the questions that you have been. We'll go as long as we can. If we don't get to your question, please email us afterwards and we'll try and get to it. So not to belabor that anymore. What about gall wasp or Japanese beetles? Do you guys take care of those directly in any way?
No, we spoke extensively with some plant pathologists from all over the country. I believe we had one that came down from Wisconsin to look at our gall wasp infestation in I want to say 2017. And she had done a lot of work with gall wasps on other species of trees. And she said, I know it looks very damaging. And of course, anytime you hear that you have an infestation in your orchard, your first reaction is I have to do something to control this. And what she had determined, and I believe she was working on her PhD at the time, was that they don't really damage the tree in a way that it would interrupt with our ability to breed with that tree. So again, just like with the Japanese beetle, it doesn't affect seed set, it doesn't affect pollen production, at least in any way that science has determined. So we don't treat them or anything like that. And I will say that the last couple of years, again, that we've just been really fortunate, again, that Mother Nature is kind of cutting us a break and not sending some of these curveballs our way. I don't know if it's due to the weather patterns in our environment or what the deal is, but we've seen a great reduction in any pests or anything like that. Mammalian pests are probably our biggest pain in Meadowview, specifically white-tailed deer. We just have regular cattle fencing around our properties, and deer they come right through and graze, especially as the buds start to swell on the trees. That's a great food source for them. We don't cull the deer via hunting them or anything like that. What we do is just make our presence known on the property by driving through. Occasionally wake our neighbors up by blowing the horn and running a herd of deer out of our progeny tests, or get out and clap my hands and that sort of thing. Just make a bunch of noise and they'll go away. But that's really about the main pests that we have to deal with.
What is the largest tree on the farm? What is it and how old is it? I guess chestnut tree. I should clarify. Chestnut tree that we've planted.
We have a nice walnut here.
Gorgeous walnuts and poplars.
I'm sure the largest by volume is probably one of the Chinese trees. We do have some very large F1s, and considering that question, I probably would have liked to have shown some pictures of the F1s we have here. Some of those are probably, some of them are 16, 18 inches in diameter and tall, and mostly have a more Chinese growth habit, but some of them are, more like an American.
What's the biggest B3F2, the most recent generation? Because they were planted in 02, and you have some that are still 18 years old.
They're probably about a foot in diameter.
Yeah,
the biggest one that I can think of.
They can grow pretty fast.
Yeah, they can grow really fast. And again, there's tremendous impact of soil type and variation. I mean, some trees that are planted within a short distance of each other and planted in the same year, they vary a lot. One may be a foot and another one 3 or 4 inches in diameter. You don't know why. It's just a microsite variations.
So we're here almost 12:30. I want to thank all of you for asking all the questions. I'm going to end with a couple here. I'm going to take 2 more, and then I think we'll wrap it up. And I want to give Lisa a chance to tell a little bit about next week's program. So one question, and as the director of restoration, I want to address this one if you don't mind. The question is: so bottom line, we shouldn't start planting trees in earnest. It sounds like the perfect tree doesn't exist yet. And so I would say, while that's true, we don't have a tree that is, that we can guarantee is highly blight resistant, and that we can guarantee has American, and that we can guarantee has diversity. Those are the 3 things that you need together. You need diversity, you need growth habit and form, and you need resistance. That package has not yet been created. While that's true, we still do a lot of planting. We do a lot of reintroduction trials to see how much resistance we can have. How much resistance is actually necessary, and we don't know that. Will intermediate resistance do the job and get this tree back into a habitat? We're pretty sure it won't do timber, but can it at least create some sort of wildlife habitat and mast while we improve the resistance on the other side and the American character? So while we aren't necessarily doing as much planting as we would do we have more resistance, there's still a lot of planting to be done. If we only planted 1%, if we planted 200,000 American chestnuts a year, it would take us 2,000 years to plant 1% of the American chestnut range. So this is a huge project. Whatever we do now, even if we ramp up tenfold, it would still be a drop in the bucket to what restoration is really going to take. So we do plant a lot. We plant a lot on mine lands. We plant a lot in the forests to try and figure out what the best way to get this tree out in the woods is and gain a lot of information about resistance and things like that. The last question that I'm going to turn over to Lisa and then have her bring it on home. Lisa, what can the public do to help with restoration besides giving money?
Sorry, I had to get back online. Well, there's a lot of volunteer activities, as we've mentioned. Once we get on the other side of this crisis, we hope you might check in with your local chapter. We have 16 chapters within the range of the species, and you can contact your local regional science coordinator. All that information is on our website. You can find pure Americans that are still wild type trees out in the forest. That's something you can maybe do safely even now. If you're on a hike and you can identify some chestnuts, and download an app called Tree Snap onto your phone and document your findings. Or as Sarah mentioned, we're trying to breed diversity into the entire restoration population. So that's something you can definitely do. We have a public comment period coming up for the deregulation of the transgenic tree. That's going to be a real 60-day effort and a campaign to make positive comments, should you so choose, to the Federal Register when that petition is posted on the Federal Register. We'd like, experts, chestnut growers to respond. We want scientific community to respond all positively towards this, because the biotech tree is a very important tool for restoration. And those are just some of the things on the top of our heads. Tell your friends. You can, again, when we get on the other side of this crisis, part of my job is getting out and about to garden clubs and local folks that have opportunities to give lectures. Sarah and I did a couple of those last year, and as recently as in March, to really share the story. We really appreciate people that share the stories. I really enjoyed hearing the stories of the wonderful team, the talented team at Meadowview today. I didn't know some of them, didn't know your story, Brandon. So that always is heartwarming to hear how deeply people feel about this tree culturally and just passionately. People really want to bring it back. So talk it up. We really are trying to market better this great story. So those are some of the things on the top of my head. And I think that's it as far as questions, Sarah. Again, a reminder for next week, May 15th at the same time, 11:30, we're going to have Chuck Lavelle. He's a musician who also happens to be a tree farmer in central Georgia. He's a great spokesman for the American Chestnut Foundation. Not only is he touring with the Rolling Stones, he manages a 2,000-acre tree plantation in central Georgia, and has some beautiful trees and pretty far south in the range. We didn't talk about Phytophthora today, but he might be part of our Phytophthora testing sites, so we can have that pathogen covered in our breeding program for resistance. So that should be a really fun one to hear his story, and then take some questions from all of you about Chuck's journey. He was supposedly he was on the bus touring with the Allman Brothers, and he was taking correspondence courses in forestry to learn how to be a forester, because his wife Rose Lane inherited this plantation, and they decided to make a go of it. And it's a hunting plantation as well as a tree farm. So should be a really fun story with Chuck Lavelle. So Anything else, Sarah, before we say goodbye to all our wonderful participants? Great turnout today, and great job, Meadowview team. You all are really our boots on the ground, talented members that they do a lot of hard work. I'll tell you that. They do a lot of very, long physical labor, especially in the summer when it's hot. We just really appreciate their work, and it's critical for our mission at Meadowview. Our flag is in the ground there for a long haul. As Eric said, we're really committed to making this mission go forward there. So thank you all. You were wonderful today. Anything else, Sarah?
I think that's it. Thanks, everybody. Hope to see you again next Friday.